Hi children, in this video I am going to give you an overview of the entire process of protein synthesis through an animated presentation. This will definitely grab your attention and will enable you to get the flow of the different events occurring during protein synthesis in the correct sequence. So please focus. Before going to the video presentation, once again a recap of the central dogma or gene expression. We see that mRNA is derived from the DNA, and that process by which the mRNA is derived from the DNA is called transcription. And the process by which the proteins is derived from the mRNA is known as translation. In eukaryotes, transcription occurs in the nucleus and translation occurs in the cytoplasm. Whereas in the prokaryotes, as we know, there is no well-defined nucleus. Both transcription and translation occurs within the cytoplasm. This difference was highlighted in the first presentation on protein synthesis. Then second difference is transcription and translation are separate processes in eukaryotes, whereas in prokaryotes transcription and translation can be coupled. Again, the third difference that was emphasized then was in the transcriptional unit of prokaryotes, the structural genes between the promoter and the terminator were many, so we refer it to as polycistronic. Whereas in eukaryotes, there is only one structural gene between the promoter and the terminator, and therefore it is referred to as monocytronic. Along with the term polycistronic, we have learned the word polycistronic and continuous. And same way for eukaryotes, when we told about, gave it with monocystronic and split. So now I brought in the three differences between the protein synthesis in eukaryotes and prokaryotes, which was already highlighted in the first presentation. So today, after the video, after visualizing all the steps involved in these two processes, I am going to give you the fourth and fifth difference, which will serve as a complexities which is confronted by the eukaryotic forms during protein synthesis. So totally in all, there are 5 differences between the protein synthesis of prokaryotes and the protein synthesis of eukaryotes. So now we are going to observe the video presentation, the animated presentation of transcription and translation. As I am presenting the video, animated video, I will be giving a running commentary so you can follow the steps in the correct sequence. So focus and visualize. We start with transcription, and DNA is present in the nucleus if it is the eukaryote, and we have the RNA polymerase, which is the enzyme that is going to bring about transcription. There are 3 stages in transcription: initiation, elongation and termination. There is a promoter on the DNA. Now the RNA polymerase binds with the sigma factor and then binds to the promoter. Once it binds with the promoter, initiation begins. Initiation begins with the uncoiling of the DNA helix, and this uncoiling is only local. Now the sigma factor is dissociated and only the RNA polymerase brings about polymerization. It adds on complementary base pairs in the 5' 3' direction on the strand which is running in the 3' 5' direction. This continues until the terminator gene is reached. Once the terminator gene is reached, the Rho factor binds to the RNA polymerase, and at this point it is terminated. The mRNA is dissociated and the RNA polymerase. This mRNA is final if it is prokaryotes, but in eukaryotes it is called heterogeneous mRNA because it contains both introns and exons. The introns are junks, they do not code for proteins, they should be removed. So they have to undergo processing. The steps involved tailing at the 3' end and capping at the 5' end, and also removal of the introns with the help of spliceosomes. See the process? Spliceosomes remove all the introns and all the exons joins together to form the final mRNA. Now this mRNA is active. It comes out through the nucleopore into the cytoplasm, ready for the next process called translation. Now this mRNA, we can see different bases. It's running the 5', 3' direction. The triplet bases are called codons. Now we have the genetic code, which is a checkerboard of codons to make reference to which codon comes to which site, or which amino acid comes to which codon. We have the mRNA with the start codon and with the stop codon. Now to start off, we have the initiation. The small ribosomal unit binds with the start codon AUG, followed by the initiator tRNA, which is bound with the start amino acid called methionine. This is called the initiator tRNA, having the anticodon UAC. It comes and binds to the start codon, and there's complementary base pairing between the anticodon and codon, that is UAC with AUG. This is the initiator tRNA. Now the large ribosomal unit comes and joins. This forms the initiation complex or the translation complex. Now on the ribosomal unit, large ribosomal unit, there are 3 sites, E, P and A. E is meant for the exit of the tRNA, P is for peptide bond formation and A is the acceptor site. Now elongation, a new tRNA reads the codon and binds with the amino acid. The binding of the amino acid with the tRNA is known as charging of tRNA or amino acylation. This tRNA, which is charged with the amino acid, comes to A site, and there is codon anticodon complementary base pairing at the A site. Now peptide bond is formed between the adjacent amino acid at P site. The ribosome shifts and the first tRNA reaches the E site to be exited. It is called the uncharged tRNA. Now the next charged tRNA comes at A site. Complementary base pairing occurs between codon and anticodon. Meanwhile, peptide bond is formed between the adjacent amino acids. New tRNA comes in the A site. Uncharged tRNA exits at the E site. Now when it reaches the stop codon, a releasing factor binds at the stop codon, releasing the polypeptide chain, tRNA, and the 2 ribosomal units dissociates. Now we have the polypeptide chain which coils to form the proteins. So I will wrap up with the last 2 differences which is going to emphasize the complexities. And we found that the complexity was found in the eukaryotic protein synthesis, not prokaryotic. So I am winding up with the 4th and 5th difference which deals with the complexities. We notice that the mRNA that is formed in eukaryote is not final. That mRNA is called hnRNA. It is not called the normal mRNA. It is the precursor of the mRNA. It has to undergo processing, and we know processing occurs at 3 steps. The 3 steps are capping at the 5' end, tailing at the 3' end, and removal of the introns by splicing. When the introns are removed, the exons are joined, and now we get the final active transcript, that is the mRNA. So this is only in the case of eukaryotes. So the first complexity encountered in eukaryotes is with respect to the transcript that is formed. It is not final. It is called the heterogeneous RNA comprising introns and exons. Introns are junks, they should be removed, and they have to undergo a process called splicing, capping and tailing, and finally they get converted into the mRNA, which is ready for translation. Whereas in prokaryotes, what is transcribed, the mRNA that is formed is final. They need not undergo this processing. So the first complexity found in eukaryotes. The next complexity is with regard to the 3 strains of RNA. Now this you must have not seen in the video, nowhere you have got a clue of the 3 strains. Which are the 3 strains of RNA? The 3 strains of RNA are, we came across mRNA, that is after transcription the product is called the mRNA. It has bases which are read in the form of codons. Then we have the tRNA. The role of the tRNA we know, it reads and brings the appropriate amino acid by binding it with the amino acid in the presence of ATP, and that process called aminoacylation. So we are familiar with mRNA and tRNA. Or if I give it in a symbolic representation, this is the mRNA and tRNA is a cloud leaf. Okay, in secondary structure. Now the third RNA is the rRNA, which nowhere in the video is mentioned. But this rRNA is very important for the synthesis of the ribosomal units. We saw the 2 ribosomal units, large and small. Okay, these 2 ribosomal units are formed by the rRNA. This is a small ribosomal unit and the large ribosomal unit. Okay, these are synthesized by the rRNA, and the importance of these ribosomal units are, these form the sites for protein synthesis, just the site for protein synthesis. And in this site itself we demarcated 3 sites. In the video there were 3 sites, E site, P site and A site. If you analyze that, E site is the site from which the uncharged tRNA exits. P site is a site where peptide bond is formed between adjacent amino acid, and A site is the acceptor site where the new tRNA, charged tRNA, comes and binds with the codon of the mRNA. There is complementary base pairing between the codon and anticodon. Okay, so that is the significance of the ribosomal units. So these 3 are the RNAs, mRNA, tRNA and rRNA are the 3 strains. Now coming to the complexity. In prokaryotes, only one RNA polymerase, see the RNA polymerase that is required to synthesize all these 3 RNAs are the same. The very same RNA polymerase is used to synthesize tRNA, rRNA and mRNA. Whereas in eukaryotes, we need 3 different RNAs, polymerases to synthesize the 3 strains of RNA. They are, easy to remember, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. So 3 different RNA polymerases are required for the synthesis of the 3 strains of RNA. Now RNA polymerase 1 synthesizes rRNA. RNA polymerase 2 synthesizes the hnRNA, or we can say the mRNA. hnRNA is found only in eukaryote because it is a heterogeneous RNA comprising introns and exons. So the precursor or the first transcript that is formed as a result of transcription is called heterogeneous RNA. Later, after its processing only, we get the final mRNA. So that is called hnRNA, and RNA polymerase 3 synthesizes the tRNA. So the difference is with respect to the RNA polymerase. In prokaryotes, only one RNA polymerase produces all 3 strains, rRNA, mRNA and tRNA. Whereas in eukaryotes, 3 different RNA polymerases synthesizes the 3 strains. For example, RNA polymerase 1 synthesizes rRNA, RNA polymerase 2 synthesizes hnRNA, and RNA polymerase 3 synthesizes the tRNA. Okay. So with this I have wrapped up the difference between prokaryotic and eukaryotic protein synthesis. Here what I have highlighted is the complexities. and it is found that the eukaryotic protein synthesis is very complicated or more complex because of these two reasons. One, the mRNA that is synthesized